Hello, I am Iona Simmons.
I'm Maya Reeves.
My name is Jayla Washington.
My name is Brenaya Bennett, and
my name is Naya Cameron.
These are my group members, and our project is titled Environmentally Isolated Microbes from a Pond.
And today I'll be giving the introduction and background for our presentation. Through its effects on pH, temperature, humidity, oxygen, and nutrient levels, the environment has an important effect on microbial growth. The combination of environmental factors in a given habitat determines which microbial species can survive, compete, and proliferate there. Basically just trying to get everybody to understand that the environment does influence microbial growth and the different types. However, each microbe or microorganism cannot survive in each environment. So each environment has its own specific species, whether it be two different species or a combination of them. Just know that every single species is not able to survive. For example, halophiles prefer salty settings, acidophiles prefer low pH settings, and etc. Now on to yeast. Basically, yeast are definitely ideal in most environments as well as bacteria, so we're just going to tap into that a little bit. Yeast are known to prefer warm, moist, and slightly acidic environments. Yeast can be knownly found in fruits, in soil, and in sugary substances. Yeast are also said to thrive in environments like bread dough and fermenting liquids such as wine, beer, alcohol, etc. However, bacteria itself is a much more broad category and can thrive in almost every environment. Bacteria can survive in extreme heat to extreme coldness, salinity, and acidic soils. Many bacteria prefers moderate conditions with neutral pH, adequate nutrients such as soil, water, humans, and animals. Basically, just to try to get everybody to understand more as well, yeast are something that deals with women as well. I know a lot of women will know about yeast, knowing that, women get yeast infections and things of that nature, and they definitely thrive in the warm and acidic environments and things of that nature, which is definitely what happens during our processes of our body with our vaginal areas. And although it is horrible, I know yeast can also help in lots of ways. Not help, but it's good in the environment of bread dough as it rises bread and things of that nature. Sometimes a lot of stuff is bad and sometimes it's good, as well as bacteria is as well. I know a lot of people think bacteria is always so bad, but some bacteria you actually need, come to find out. So finally, the importance of isolating and studying microbes from different environments helps scientists better comprehend microbial variety, adaptability, and ecological roles by isolating and analyzing microbes from different habitats. Additionally, it results in discoveries that can have issues in the real world. All in all, researching microbes that live in harsh environments can provide insights into survival strategies, environmental conditions, and climate change. So, to end off on my slide, basically I just wanted to give examples of the different ways of microbials and how they can survive in each environment. I know that the importance of isolating and studying microbes from different environments can definitely help us understand the microbial diversity, adaptability, and ecological roles better. Because in a lot of ways, a lot of people would think, oh, this low pH can go inside of a different environment, and it's not able to go there because that environment uses high pH levels. So just making sure we understand everything will help us better as humans to have better biotechnological tools, and basically to help us understand that studying can reveal mechanisms of survival that may inform fields like astrobiology and different forms of environmental remediation.
Our hypothesis is that the pond water samples has an effect on the amount and the type of bacterial growth. This meaning the water samples has an influence on both how much bacteria grows and which type of bacteria grows. What does that mean? Different ponds can carry different nutrients, pH, and have different temperatures and other environmental factors. So if the pond has too many nutrients, some bacteria may die. So this is very important when you're trying to figure out which bacterium, what do they need. By observing bacterial growth from each sample, we can learn how specific water conditions shape microbial communities. This can help us understand ecosystem health and microbial diversity in different aquatic environments.
I'm going to be presenting my slide 1 method. So the experimental design. In this experiment, microbes were isolated from pond water near the engineering building. Pond water samples were collected using sturdy tools and transported to the lab. The water streamed onto nutrient agar plates using sturdy technique to avoid contamination. The plates were then left at room temperature for 1 week to encourage bacterial growth. After the colonies developed, 2 distinct bacterial colonies were selected for gram staining. The experimental location. The pond near the engineering building was chosen because standing water is a natural reservoir for diverse microbial communities. Ponds often contain bacteria from soil, plants, animals, and other environmental sources, providing a rich and varied sample.
And now we're going to move on to the results section. So first we see figure 1, the agar plate sample 1. And as you can see towards the left of the agar plate, there is a lot of bacteria growth. If you look closely towards the left and in the middle, there is a different bacterium type. And towards the right of it, you can see that there is bacteria growth, but it is more sporadic. Okay, so figure 2, the agar plate sample 2. Towards the left, you see a lot of bacteria growth, a lot of round shape bacterium, and it is close and compacted with each other. Towards the left, we do see some bacteria, but it is, like I said, more sporadic. And in the middle of the agar plate, you see one specific bacterium type that is different from the rest that I will address later. Okay, so for the experiment group, the pond sample number 1, the number of CFs used was 75, and the number of distinct bacterium types was 2, and the bacterial morphology was punctiform, which is pinpoint, and it was also round. For experiment group pond sample number 2, it was too many colonies to count. So, and then the number of bacterium types were 3, and then the bacterial morphology was punctiform or pinpoint, and it was irregular and round. Okay, so moving on to the graphs. The first graph shows the number of colony forming units or CFUs for each sample. Pond sample number 1 had 75 CSUs, while sample number 2 had too many to count. So we estimated about 150 to 175 and labeled it as TMC. This suggests sample 2 has significantly more bacterial growth. The second graph displays the number of distinct bacterium types found in each sample. Sample number 1 had 2 different types, while sample 2 had 3, showing greater bacterial activity diversity in sample 2. The third graph is a stacked bar chart showing bacterial morphologies. So sample 1 showed 2 types, which is punctiform and round, and then sample 2 showed 3 types, which is punctiform, irregular, and round. This tells us that not only did sample 2 have more bacteria, but they also varied more in shape. When we looked at the samples under the microscope, sample 1 appeared pink, indicating that it is gram negative. Sample 2, on the other hand, appeared purple, meaning that it is gram positive. This shows that the bacteria in each sample differ in their cell wall structure, which may affect how they respond to antibiotics to survive in their environment, essentially.
And I will be presenting the discussion portion of our presentation. Pond water sample 1 produced about 75 colonies with 2 distinct morphologies, pinpoint and round, while pond water sample 2 was so densely covered that individual colonies could not be counted and displayed 3 distinct morphologies, pinpoint, irregular, and round. This contrast shows that areas with richer nutrients support both more bacteria and a wider variety of colonies. Plate counts can be inaccurate when pipettes aren't precise, agar layers vary, or plates become overcrowded. So to improve this accuracy, we can calibrate pipettes and run dilution in sets of 3, keep agar depth and spreading consistent, and dilute further until the plates can hold about 30 to 300 colonies. Working in a sterile hood with negative controls and using image analysis software to count the colonies will also reduce error. Because health agencies rely on these counts to flag pollution and pathogen risks, these adjustments will help us get faster, more reliable warnings, and ultimately help protect water quality and public health overall.